It's my honor to be here and to have this opportunity to exchange and to share PRRS experience with scientists and scholars working on PRRS and PRRS virus. Also, I would like to thank the organizing committee, also and Dr. Bob Rowland to invite me to be here. So my talk will focus on pathogenesis and control of highly pathogenic PRRS virus in China. So most of you know, 10 years ago, a highly pathogenic PRRS virus, we call HP-PRRRS virus, emerged and prevailed in China. Also, the virus spread to the neighboring countries such as Vietnam and Philippines and Laos, probably also the North Korea. So this virus is displayed high virulence for pigs, including sows and green pigs, which caused high fever, high mobility, and high mortality. Also for the affected pig herds and sows, the virus caused severe diseases, so including severe abortions in sows and severe respiratory disorders in green pigs. My presentation will focus on following points, including pathological pathogenesis and molecular basis, and immunosuppression, and the evolution and diversity of PRRS virus in China. And also, I'm going to mention a little bit about control of HP-PRRS virus in China. But my presentation do not include all the experts related to HP-PRRS virus, because a lot of research teams are working on HP-PRRS virus. So they got a lot of advances and progresses on this virus. So my presentation just mentioned some important issues or important progress. So point one is regarding pathological pathogenesis of HP-PRRS virus. This virus infection can cause acute lung injuries. When the virus inoculated the pigs, so the virus can cause the most serious lung injuries, including severe pathologic lung lesions. So we can see the lungs showed increased neutrophils, mast cells, and mononuclears. Also, the lungs showed severe fibrosis. So this is our team research work, which is published in Veterinary Microbiology. The second, regarding pathological pathogenesis of HP-PRRS virus, we found HP-PRRS virus can be detected from more tissues by immunohistopathological chemistry. Also, in these tissues, the virus showed 10 and 1,000-fold higher viral loads compared with no pathogenic PRRS virus. From this tab, you can see we can detect HP-PRRS virus in some tissues, but we could not detect the PRRS virus in these tissues. Also, the positive rat showed some differences between HP-PRRS virus and no pathogenic PRRS virus. Also, many research work found HP-PRRS virus showed a higher replication efficiency in the affected pigs. This is a study from another research team in China. They found HP-PRRS virus, that's a strain HUN4. The virus can infect the alveolar epithelial cells, macrophages, and vascular endothelial cells, which can cause diffuse alveolar damages and lymph node necrosis. Also, this virus showed a higher replication rate in these infected tissues. So point 2, regarding molecular basis of HP-PRRS virus, virulence. So HP-PRRS virus is a model virus for investigating the molecular basis related to the virulence. So in 2009, our publications reported our data regarding the HP-PRRS virus. The virus displayed a fatal virulence for piglets. As most of you know, the NSP2 coding region has totally 30 amino acid deletions. This is molecular markers for the emerged HP-PRRS virus, but our research work found this deletion is not related to its fatal virulence. After we use the infections clones to exchange the NSP2 regions between highly pathogenic PRRS virus and a low pathogenic PRRS virus, our work need to further which regions are related to the virulence of HP-PRRS virus. So we continue to use infection clones to construct the different chimerical virus between HP-PRRS virus and no pathogenic PRRS virus. We constructed more than 12 chimerical virus, and we analyzed their pathogenesis according to the clinical signs and gross non-lesions and macroscopic lesions, and also mortality of the affected pigs. So our data, the slides has changed. Our data showed NSP9 and NSP10 are related to the replication efficiency in vive, and also we concluded both of them contributed to the fatal valence of HP-PRRS virus, because our experiment found when we exchange the NSP9 and NSP2, replace the NSP2 and NSP10 by the low pathogenic PRRS virus, so the chimerical virus, the virulence declined. But we replaced the NSP9 and NSP10 of no pathogenic PRRS virus by highly pathogenic PRRS virus, we found that the virulence increased. So we got these conclusions. The point 3, regarding immunosuppressive aspects of HP-PRRS virus. As we know, the PRRS virus infection can induce immunosuppressions. So many research teams focused on the inflammatory cytokines induced by HP-PRRS virus, including some research team in China and also the research team in the United States. So they published many papers to describe this phenomenon. Also, they found there are differential expression of these cytokines between HP-PRRS virus and no pathogenic PRRS virus. Their work involved in IR1, IR6, and TNF-alpha, also including IR10. So some research got a conclusion. So HP-PRRS virus can induce cytokine storms. So because of time limits, I do not want to give you more details regarding this research. Another interesting finding was done by another research team in China. They found HP-PRRS virus can cause thermus atrophy and the deletion of thermocytes of pigs. They use HP-PRRS virus infected piglets, and they found the virus can induce higher thermocyte apoptosis. Also, they found most of the apoptosis cells were CD3 positive T cells, but these cells are not infected by PRRS virus. They can only detect the HP-PRRS virus in CD14 positive cells. But what's the link? What's the mechanism? The HP-PRRS virus can induce the thermus atrophy and the depletion of thermocytes. This is a need to be done to do more works. Also, in our lab, we found HP-PRRS virus can downregulate MHC1 molecular expression on palms. We found HP-PRRS virus can significantly induce the MHC1 expression on primary macrophages. We found that HP-PRRS virus can target MHC1 degradation by ubiquitin proteasome systems. Also, we further found the viral protein NSP1 alpha contributes to this degradation. And also we found NSP alpha interacts with both chains of MHC1, including the alpha chain, the heavy chain, and beta 2 microglobulins. We also found structure integrator of NSP1 alpha is required to induce MHC1 molecular degradations. So this fund can suggest HP-PRRS virus infection can induce this immunosuppression of the cellular mediated immunity. So this is our recent work, which was published on veterinary microbiology. We found HP-PRRS virus could suppress Th17 cell response in vivo. We use a monoclonal against the Th17A cytokines to monitor the change of the peripheral blood and the lungs, where we found the HP-PRRS virus infection can suppress Th17 cell response, also can cause a reduced Th17 cells in lungs. Also, we found HP-PRRS virus, they can significantly induce the higher bacterial loads in lungs, because we compared the bacterial loads in lungs of the affected piglets between low pathogenic and highly pathogenic PRRS virus. But we do not know the link, the mechanism between the Th17 cells and immunosuppression induced by HP-PRRS virus. Also, this is our future works. Okay. So point 5, I'm going to mention a little bit about evolution of and diversity of PRRS virus in China. So after the emergence of HP-PRRS virus, the virus continued to evolve, continued to undergo the genetic variations. So as most of you know, in China, the majority of PRRS virus belong to genotype 2 PRRS virus. However, we have multiple lineages. We have multiple subgroups. Probably we have more clades, because in our lab we monitored the variation, the evaluation of PRRS virus in China. We detected more samples from the different pig farms. Also, we have HP-PRRS virus. We have low pathogenic PRRS virus. We have field virus. We have vaccine virus. Also, we have recombinant virus among field virus, or between field virus and vaccine virus. Good. This figure shows you we have various strain of PRRS virus with different deletion or insertion patterns in NSP2, because we analyze the NSP2 from the gene banks available. All the strains belongs to Chinese PRRS virus. So we found at least 23 patterns of NSP2 deletions or insertions. So we have so divergent strain of PRRS virus. Also, we have so many different patterns in their NSP2 genes. Okay, next. Okay, I'm going to mention a novel strain of emerged in recent years in China. We call the NIDC30-like virus. This is just emergent and prevalent in China, because you can see the CHSX1401 we isolated. They showed a high homology with NIDC30. This virus just described by Dr. K. Fabre's research team. in 2012. So we placed these isolates just in the tree. You can see very close to NIDC 30 in the United States. Also, the virus showed as identical deletion to the NIDC 30 in its NSP2 codon regions, totally 131 amino acid deletion. The deletion pattern is 111 plus 1 amino acids and plus 19 amino acids. So totally 131, which is identical to the NIDC 30. So I do not know the origin or source of this virus, but I just speculate this virus can be introduced by breeding pigs, probably from the United States. I don't know the origin. Also, the virus was involved from NIDC 30 isolates in 2008 in the United States. Now the virus provided in many pig producer areas in China. The virus can cause abortion of pregnant sows and respiratory disorders in growing pigs in many pig farms. The virus belongs to be virulent, but less virulence compared with HP-PRRS virus. Also, a lot of research team found a virus which has undergone genetic exchange with the HP-PRRS virus. That means the virus is easily recombinant with the feeder virus, with the HP-PRRS virus. Very easy. Also next, my lab also found a virus which is a recombinant virus between HP-PRRS virus vaccine on the pig fields and the pig farm, and NIDC 30-like virus, but this is not published there. Okay, we move to the final points regarding the control of HP-PRRS virus infections. So PRRS is still a major challenge. It's the major issue we have to face in our surveying industry. So to control PRRS in China, the vaccination is a major. So here in this table, we just list the commercial HP-PRRS virus-derived MRV vaccine in China. We totally have 4 strains of HP derived from HP-PRRS virus. So since 2011, we have these commercial vaccines which are used on pig farms. They can provide an efficiency protection against the feared HP-PRRS virus, but we have noticed the safety concerns on the pig farms. We need to see the massive vaccination on pig farms using these vaccines clearly have benefited the reduction of outbreak and the incidence of HP-PRRS in China. However, there are many problems related to the use of these vaccines, including reproductive failures in vaccinated sows, such as sporadic abortion and increased stillbirth rate, increased weak piglets, also including respiratory conditions in the vaccinated growing pigs. We have observed the increased PRRS morbidity and mortality in nursery pigs and finishing pigs. Also, we found that the vaccine virus can circulate and spread on pig farms. Okay, next. This just I show you this pigs with pig farm with a vaccination of HP-PRRS virus derived MRV vax showed colitic PRRS. Because I visited many pig farms after vaccination using HP-PRRS virus derived MRV vaccines, we also observed many problems, including more than 5% pregnant sows suffered abortion following vaccination using this vaccine. Also including increased stillbirth rate. And natural pigs showed colitic PRRS 2 or 3 weeks following vaccination with HP-PRRS vaccines. Also poor growth performance of pig herds in nursery and feeding pigs. Also about 20 to 30% morbidity and 10 to 20% mortality in grown pigs. We detected the virus. We detected the genes of PRRS virus from this pig, from these sick pigs, which showing the colitic PRRS, and sequenced the genes. We found the virus very similar closely to vaccine virus. This is the data from another research team, the Dr. Guangzi Tong's research team. They published their data to find some field strain are likely to be revertant of the vaccine strain JX1 passage 80. This is MRV derived from HP-PRRS virus. Also in my lab, we have many data, or just reporting in my lab. So this is the problem we have. 5 minutes, okay. I noticed the clock. So in China, we have many challenges facing the control of PRRS. The first is diversity of PRRS virus strains. We have so many PRRS virus strains with different genetic characteristics. We have different pathogenesis. Also, the virus continue to evolve, and new novel strains will continue to be emerging. Also, we have so many vaccines. How to choose this vaccine to control PRRS? This is a problem that for pig producer have to face. How to strategically use HP-PRRS virus derived MRV vaccine. We need to restrict it to use. We need to stop use. Okay, next. So this is our presentation. I just gave some summary. So HP-PRRS virus infection caused severe lung lesions and expanded tissue lesions of pigs. HP-PRRS virus infection induced the cytokine storm. HP-PRRS virus has stronger ability of suppressing cell-mediated immunity of pigs. Within its genome, NSP9 and NSP10 together contribute to the fatal valence of HP-PRRS virus. High replication efficiency in vibe of the HP-PRRS virus contributes to its high pathogenicity. Evolution of diversity of a PRRS virus is a great challenge facing the control of the PRRS in China. Also, we need to do more work. We need to do enough work. So, future work will be done to understand the molecular pathogenesis of HP-PRRS, such as is there any amino acids related to the virulence of HP-PRRS virus? we are going to do this work. Also, reasonable strategical use HP-PRRS virus-derived mRNA vaccine. We need to think about that. We need to think about our vaccination strategies. So how to do that? We also need to push forward the elimination of PRRS virus on breeding pig farm to construct a PRRS selective breeding herds and farm. We also need to push out implement regional elimination of PRRS virus in China. So I'm happy in recent years our Chinese CADC initiated a project on making authentication for disease eliminating breeding pig farms, including the collected swine fever and short rabies and PRRS. This is our future directions to control PRRS and other swine diseases. Okay, I'd like to thank the foundings, also my research teams. Also, I'd like to thank all of you for your attention. Thank you.